Good morning, sir.
Very good morning.
Good morning, sir. Respected Professor R.K. Goyal, sir, Hon'ble Vice Chancellor of Delhi Pharmaceutical Sciences and Research University, Delhi. And I'm very happy to have you in this session to chair the session. You are a leading academician, researcher, and you have so many facets, and you are now dealing the one and first University for Pharmaceutical Sciences in India. There is Delhi Pharmaceutical Sciences University, and we are very happy to have you in this platform to chair the session of Professor KPR Chowdary. And so Professor KPR Chowdary is one of the senior professors of pharmacy in India, having more than 44 years of experience, and he was the former principal of Andhra University College of Pharmacy, and now he is the research director in because Institute of Pharmaceutical Sciences, Rajahmundry, Andhra Pradesh. So I'd like to welcome Professor KPR Chowdary. He is continuously serving to the scientific community, and every scientific publisher in Indian Pharmaceutical Congress will know very well about Professor KPR Chowdary. He is running the scientific show for the past 3-4 IPCs, and we welcome Professor KPR Chowdary on this platform of APC Online Convention to give a lecture on research methodologies in pharmacy in objective and explanatory studies, so experimental studies. So without wasting or without taking much of your time, I would like to welcome Professor R.K. Goyal to take over the session and chair the session. I request Professor Goyal sir to chair the session. Welcome, sir. Now it's
okay. Thank you very much, Dr. It is a real pleasure for me to chair the session of KPR, Professor KPR Chowdary. And by that, you have already, I will say you have obliged me by getting me this platform. I'm very much thankful to the Alwar Pharmacy College as well as the Rajasthan Health University Sciences. Now, I know Professor Chowdary for a long time, let me say. I remember 2004 when you have organized the conference in the Vizag and APTI convention, and there I was given the charge of. Can you allow me to share the screen?
yes, sir. Please, we are pleased to.
Okay. So I remember that.
Yes, sir. You may screen share.
Okay. So at that time, I remember I was given the Best Pharmacy Teacher Award. I was the real fortunate to have this award. And since then, I have been more closely Professor Chowdary in APTI also. You are talking about the IPCs, but in APTI also, he has done substantial work, I can say. And has been awarded with the most prestigious awards. And I know what he is. Now, just to introduce the subject, let me have, I'm sharing, can you see my screen?
Yes, sir, we are seeing. You make it as a screenshot.
Yeah, okay. So what I'm going to do is I will, okay. Okay, it's not coming. Okay, anyway, Now, I just to introduce the topic, very much important. We all know what this corona is playing the havoc. The numbers are increasing day by day. This was just 2 or 3 days before the scenario in the world. Now it is 12 lakhs, or sorry, 1 crore and more than 2 lakhs, it has crossed that. 12 million now. And then when it comes to 120 million, and then come to India, again, very bad situation. Well, under these circumstances, in the pharmacy colleges, what to do? That's what is the topic which Professor K.P.R. Chowdary is going to do. But when we think of this, I'm just trying to slightly correlate what can be done. And of course, it will be Dr. K.P.R. Chowdary to give more. That we always think of a new drug, and this is very much needed in the COVID time. New drug, how to treat. That is the unrevealing certain mysteries of pathophysiology, what this COVID virus is doing to the world, and something related to pharmaceutics or the quality assurance and all. That is what is a challenge already there. We all know that Indian pharma has done the best. I will not go into the number. It is all well known. Day by day, the numbers are increasing. In terms of medical devices or masks, which we were the lowest, we have suddenly gone up. That is also there. The several transformations are occurring. We are truly now international. Even the US and all, they have surrendered that no, India is better in the pharma. That's what has rapidly expanded to this level. About the pharma, the main thing is the greatest risk. Everybody is talking of the vaccines also. But the problem is, will vaccine come within no time? The answers are lying here in this slide also. It is everybody expecting safe, but fast. Now, safety and fast cannot come to clinical research. That's what is again the problem. So, in this case, and then we have to still think of the quality of research and patents and all that. Now, in this era, everything is open. The journals for which we used to struggle, we have started getting free, especially if it is for COVID. This is what is there. With the idea, I will just have only 4 points to say. If it is a pharmaceutics, nanotech and biotech and biosimilars, yes, you have to think thoroughly in that. When it comes to pharmacology, biomarker research, translational research, and medical devices, they have to be considered. And biomedical, translational, I'm talking, yes, our Ayurveda, the wealth can be translated into the COVID fighting. When it comes to chemistry, yes, based on docking, this is what I also projected to the government, and they have taken in the hackathon also the same concept. Yes, have the compounds studied in silico and then go ahead. This is what is there. So this is how ultimately the drug has to come from the herbals. That's what is doing. And finally, the management. Yes, management, very important. Pharmacoepidemiology, epidemiology as such, how the trends are going. All these things will come. And finally, of course, with these words, I will simply say that there are a lot of things to be done. The public has to be made aware about that. And based on that, we have to go ahead. So, it's a very, very important topic. That's what I'm trying to tell. And so, I will not take more time. I have just tried to introduce in brief the topic, and over to you, sir. I think we will not waste more time in introducing, but you are a personality in the world of pharmaceutical sciences. Dr. Chowdary, sir.
I would like to request Dr. K.P.R. Chowdary to. Yeah, please.
Thank you. Thank you. Now, Honorable Vice Chancellor, sir, and Principal of the Alwar College of Pharmacy, Dr. Jaybalan, and learned faculty members and prospective students and research scholars attending this seminar. And good morning to all of you. First of all, I congratulate the Alwar College of Pharmacy, the management and the principal for the excellent initiation they have taken in conducting a convention, APC convention online, and started this convention. I understand that series 1 is already over, started somewhere in May and extended to June, and as many as I understand 18 to 20 sessions are being held under series 1. And the series 2 also started this month beginning on 6th probably, and they are arranging continuously one or other lectures for the benefit of the pharmacists working in various sectors, settings, as well as mainly the faculty of various pharmacy colleges and the students. And when Dr. Jaybalan approached me over phone, I readily accepted, because I'll take always pleasure in interacting with the youngsters and to some extent motivating them towards what is needed. And I am very much fortunate today to have Dr. Goyal Saab, the Vice Chancellor of Delhi Institute of Pharmaceutical Sciences University, a very famous establishment in India, and as the chairperson, and I feel that I am really honored to have him as the chairperson for my session. And I also thank Dr. Jaybalan for the opportunity he has provided to me to interact with the faculty and students and others of various pharmacy colleges through this webinar. And Professor Goyal Saab already narrated in his introductory remarks the need for research and in which way we have to move in terms of research, beautifully subject-wise in pharmaceuticals, formulations, pharmacology, pharmacy practice, biotechnology, vaccines, etc. Item-wise, SAR has already identified the need for conducting extensive research in pharmacy. And research, the word research is there since long. But the question is whether we are doing meaningful research or not, effective research or not. What is that effective, meaningful research? And in the academics, the research component is introduced in the PG programs, means the MPharm and PhD programs long back, starting in 60s itself. The concept of doing project work, research work fully for either 6 months for some time or 1 year for a long time is introduced. So all colleges and teachers, principal, certainly they are involved in doing some sort of research. But are they resulted in any new findings? Because the object of research is to generate new ideas, to generate new knowledge. With respect to pharmacy, certainly it should generate new drug molecules or improved molecules and alternatives for treating various diseases and disorders, etc. And excellent progress is there in India as far as the pharma industry is considered, and the research carried out by the industrial pharmacists. Everything is extraordinary. That is the reason why today the entire world is looking at India only for supplying a vaccine for the corona, COVID-19. And we are sure that soon India will produce an effective vaccine for COVID-19 definitely. And but this is all the pharmacists, pharmaceutical scientists doing in industry. But to be frank, to what extent the academicians are doing is very unfortunate that the academicians in the bulk of the college. So there are a few institutions like DPSR, since long, DPSR and such other established universities, central universities, and established state universities, etc. They are doing good. But whereas now when you consider the pharmacy, there are so many self-financing colleges, private colleges otherwise, and you consider the state of affairs in all these colleges, whether they are doing any sort of research or not. And hence it is high time that all of us should motivate the managements first, the faculty and the students to show more interest in the research. And Jayawal, may I have the first slide, please? May I have the first slide, please? First slide, please. Okay, thank you. Thank you. So that is why it is called research and its importance. And in industry, they are doing excellent. And in academics also, research is also important. What is research? We already seen briefly. That is, research is a scientific, well-planned investigation or a series of investigations to generate new knowledge. That has good application and to serve some purpose in the society. And hence the industry, especially pharma industry, is always a research-based industry. And the pharma industry, the research that has been done is always application-oriented and new, and that is the reason why many patents in the pharmacy are from industry only. The patents from academic institutions are very, very meager. But in academics, is it necessary to take up the research? Yes, certainly it is necessary, because the academic institution, faculty, teachers' responsibility is to train, essentially to train the students. And apart from the teaching, research also forms an important component. In fact, the research and teaching are considered as two eyes of an individual. Both are equally important. But as far as the teaching is considered, so almost all institutions that are approved by the regulating bodies like ACT, PCI, we can expect that they are up to mark in teaching and learning process. But when it comes to the research, many are neglecting. Many are neglecting. And this is evident in the government annually gives the NIRF rank. So every year in the month of February or March, based upon the information supplied by the institutions only, assessing the various achievements of the college, and the ranking will be given. And in India, there are as many as 3,300 pharmacy institutions, total of government as well as private sector, out of which only 100 institutions are shown as those qualified in the first 100 ranks. They are recognized only the first 100 ranks. So the remaining bulk, they are not. And when an analysis is done, why several other institutions are not getting the minimum score, the reason is they are totally ignoring the research. With regard to the other things like the teaching learning components and student success, employment, everything is good. But when it comes to the research component is much neglected in the institutions. And research component is very important for teachers to their progress as well as to get credit to their institution. So everywhere, when a teacher is evaluated, I think all of you might have attended several interviews for selections as well as for ratification by the universities. And many times it is only the research contributions will be asked, but not the teaching. Of course, teaching, only measure of teaching is that because it is only the student success. And today, the student success is at a higher rate because of various reasons. But research is very poor. Research is counted by the number of sound publications in reputed journals of high impact recognition, as well as by the number of patents that a teacher or an institution is getting. And also the projects conducted with the government support or industry support. It's not that the management can earmark some amount for the teachers to do the research, but teachers should come up to the level to grab there are so many funding agencies supported by the government to get these funds and execute, and those funds only will be counted, not, and also they should impress the industry. That means collaborative research with industry is very much needed, then only the quality of research will improve. And today what in 90% of the college is going on is only a routine type of research. When one student 2 years back did some work with drug X, the same type of work will be repeated after 2 years with drug Y. And there is no charm and there is no positive result, originality. Such researches will never be considered. And hence, all the academicians in the institution should think about how to do effective research. So for doing effective research, certainly the concept of research and requirements of research or good practices of research are to be clearly understood by not only the students but also by the faculty. And hence, when I thought of speaking on the research, and I have chosen to speak on the basic concepts or principles of research, methodologies, and how the research methods can be organized to get the unbiased result. That is one of the requirements, reliable result. So because the outcome entirely depends upon how the experiment is conducted and how the response is measured and how it is estimated or interpreted like that. And hence, may I have the second slide? Next slide, please. Next slide. So this is the research in pharmacy. Research in pharmacy. Areas of interest first we will consider. As already pointed out by Dr. Goyal Saab, so the research all is related to drug discovery and development. So the major research needed in pharmacy is to generate newer drugs, and hence drug development, drug discovery and development forms the major part of the research. And we all know that the major source of the drugs is organic synthesis, and hence we need a strong foundation in the principles of organic chemistry, basing upon which we can aim at designing newer and newer drug molecules. These all results in chemical drugs. And even today, the organic synthesis chemical drugs form the major source for drugs. And in the recent past means in the last 2-3 decades, other type of compounds, other type of drugs known as the biologicals, which are the protein and peptide, large molecular drugs and enzymes particularly wonderful identified, and they are all very considered as very life-saving drugs. And though vaccines are very old, but still it's a continuing the demand. Like this, in the biologicals, these are otherwise produced by, earlier it is known as the fermentation technology, now it is called the biotechnological means, and hence we have to improve our knowledge and skills in conducting the biotechnological research to generate newer and newer biologicals. And the age-old area is known as the phytochemistry and herbal drugs. And since long, these are in usage. Several wonderful molecules are derived from natural sources, particularly plants and herbs, and they are doing well. But with increased awareness in the public, now evidence-based medicine is the need of the hour. Whatever the doctor and the pharmacist suggest as a medicine for curing a disease, we should also create an evidence. And unfortunately, though several medicinal plants, herbs are available, and they are claimed to have several effects, beneficial benefits, But so far, as the evidence is considered, no scientific validation. And hence, in this area also, there is a scope for conducting a lot of research work, original research work, particularly for developing the evidence, and hence for quality control that is also missing for all other chemical and biological type of products. They are well developed quality control, quality assurance techniques. But for herbals, the techniques are still needed, and hence it is another area where the faculty and the researchers in academics can also concentrate. And later on, these are basically the essential, but you have the extension of that, the various aspects in the drug development process, like NC characterization. A lot of research is needed, instrumental methods are to be used to characterize various properties, physical properties. Similarly, the analytical method development is a continuous, always there is a demand that uh, for all the drugs, formulations in the drug in the preparation, drug in the biologicals. So for ADME studies, for bioequivalence studies, for stability studies, analytical method is the most important one. And hence the analytical method development. So many methods, knowledge has improved in this area, and there is still a lot of scope for developing newer and newer methods. But newer and newer methods, okay, but always what I observed with different faculty is that they concentrate only on using a method like HPLC, though it is good, but it now has become again obsolete, and it is not suitable for estimating the drugs and metabolites in ADME studies, drugs and metabolites in pharmacokinetic studies, in biological fluids. So to go beyond. And so in the institutions, always the limit is fixed as HPLC only, but that too HPLC methods are applied in the API analysis as well as the formulation analysis, but not going beyond that. Pre-formulation and formulation development. So this is normally a topic, a wonderful topic for the industrial pharmacist. A lot of weightage is given to this because with formulation only the industry can think about doing the novel things, and patentability can be achieved for a new drug easily, and the life of a drug can be extended. And because of such positive aspects, the formulation development forms an important activity in the industry. The same way, it can also be introduced into the academic institutions. Certainly, they are doing, we are doing. But think above the laboratory. So here you have to interact with industry. You have to interact with industry and then take the support from industry, or establish research institutions, and then some sort of research you have to generate. So certainly the one area Sar is also referred as nanoparticles. Certainly it's a wonderful area, nanoparticles. Okay. And for doing a nanoparticle research, so we have to first make an assessment whether an institution has the at least minimum facilities to do the nanoparticles. To preparation, more important is stabilization and isolation as nanoparticles, and formulation. And hence, for all these aspects, definitely it should have instruments like freeze dryer, okay, or spray dryer is needed to convert the nano suspension into particles, nanoparticles, and then finally compress into tablets or caps, because these are the ultimate dosage forms more widely used. So in the same way, identifying the area, and the immediate job of the management and the principal is to create the suitable infrastructure. And without infrastructure, you cannot aim at doing good quality research. That is the minimum first requirement. And similarly, process development, stability testing. May I have the next slide, please? Next slide. Stability testing, next. Okay. And preclinical, that's well developed. So animal studies for assessing the various pharmacological effects, whether it is therapeutic or toxic, toxicological studies, ADME studies. So extensive work is needed in all these areas, which form an important area of research that can be done even in the academics also. And clinical studies. So clinical studies, you all know that phase 1, phase 2, phase 3, pharmacovigilance is coming up now. And clinical evaluation of the prescription treatments, that is pharmacy practice. With the introduction of the pharmacy course in the country, now there is a good rapport happened between the academia and the hospital. And wherein the hospital means it is involving the patient, treating the patients with various treatments, drugs, and certainly we should be able to identify, evaluate whether the prescription is serving the purpose or not, and outcome research, how it is influencing the result, etc. And all these are good areas of research for the pharmacy teachers and students. Next. So then we think overall the steps in research investigation, whatever is the type of research, whether it is related to nanoparticle, whether it is related to development of vaccine or formulation, any other formulation, or a phytopharmaceutical, whatever it is, there are some things which you have to clearly understood, the steps. So the first is a statement of objectives based on a survey of the existing literature. That's very, very important. So the first requirement for any successful research is a literature survey. Literature survey means the person who is doing the research should become competent with the knowledge as well as skills in that subject. Okay, without having a knowledge and skill in a particular subject, there is no point in taking up research activity. So the objectives should be clearly defined. So objectives, how the objectives are defined is by making a thorough survey of the literature. You understand the knowledge, the current status of knowledge nationally, internationally, summarize and understand thoroughly. And from that, you come up, search the areas where we can further do investigations that will generate in the new knowledge. And hence identify those areas and develop the objectives based upon that. And hence the first part is that literature survey, and if it is done properly, of course sometimes it will be continuous. Even after starting the project also, you should continuously go on making survey and updating our knowledge in a particular area. Then literature survey, basing upon that, objectives are to be clearly defined. When once the objective is clear, the project is considered as halfway successful. And the main reason given for failure of many of the projects, even by the eminent personalities, is that they don't have the clear-cut idea what to do in the beginning. If they have the clear-cut idea, then certainly the result is success. And then write the procedure for randomization. That is what is known as normally whatever is the research, it include two things.
Excuse me, participants. This is something happened with the internet connectivity of Professor KPR Chowdary. Hope he'll be connecting soon back with us. I'll ask him to.
In the meantime, if there are any questions, if they can ask.
Sir, there is no questions in the question and answer section
Okay.
so far. I'll try to connect Professor Chowdary and inform them that his internet connectivity is failed. So we are live on, sir. Professor Chowdary joined now. There is a disturbance of internet and power connectivity. So please, Chowdary sir, please unmute yourself and continue.
Hello?
Yes, sir. You are audible.
Hello,
Now.
is it clear? Is it audible?
Yes, sir.
Audible,
You are audible, sir. You are audible. Please carry on, sir.
Okay. So sorry for the disturbance in this area. Now power supply stopped. So internet failure. Now connected to the mobile net and hotspot. Now that is in the procedure. Now what you have to do is decide on the number of replications. So in research, the sufficient number of times the same experiment has to be conducted. Otherwise, it is not taken granted with one or two observations. We will get the correct value as expected. Now, the statistics will provide several ways and means of determining the sample size or number of replications. A bit later, we will see. And always we have to include a control group. Without the treatment or without the drug, or sometimes a placebo is given, a similar product without active drug, and hence this should be always taken into account, especially in pharmacy when we are making evaluation of the drug effects. So there may be some natural cure, and hence one group of subjects or animals should be kept control, and whatever result is obtained in both the groups, control and drug treated groups, are to be compared by various means. And next slide, please. Next. Next.
Yes, sir.
Next. No, not moved. Okay, okay. And then define and use objective measurements of responses. That is more important. Many times we have to avoid better to avoid subjective responses, objective responses which can be quantified or which can be counted as numbers will serve the purpose. And measurements or estimates responses. So for that, we have to use always the scientifically accepted methods only. Scientifically accepted, the methods are to be again validated. So the concept of validation is utmost important in research, not only in industry. In research also it is important. Whatever equipment or materials or methods we use in research, that should be validated and confirmed that they are working as expected with good accuracy and precision. Then only such things can be used in the methodologies. Then similarly, identify the statistical methods used for comparisons and for making the conclusions at the end. Then finally, act on the results appropriately by either by publication or by patent research, etc. Next slide, please. Okay. So now with this background of the importance of research and how it is normally done. So there are certain good research practices, just as good laboratory practices, good clinical practices, there are also good research practices. That whatever is the type of research we are doing, we should make use of them. The principal one is absence of bias. So bias means giving a certain undue weightage or favorism to one treatment, one drug or one product like that. And why you should avoid this bias? If bias is there, the rules of probability are not applicable. And hence, to enable us to apply the rules of probability, this bias should not be there. So, unbiased way, in an unbiased manner, the experiment has to be conducted. And just I will give a simple example how important the concept of bias in calculating the probability. Suppose all of us know when we toss a coin, there are two possibilities. So probability of getting head is half. Suppose that is unbiased. If you take only unbiased coin, if the coin is biased, it can be biased by applying a coating with magnetic material. Then if you toss it, that coated surface will always go down and other uncoated surface will turn out, and hence the probability of half is not applicable. The probability changes. So in the same manner, whatever experiment you do, to enable us to apply the probability. Why the concept of probability is important? Because all the statistical methods are based upon the concept of probability only, and hence the experiment should be conducted in an unbiased manner. Then only the statistical methods can be applied and inference can be made correctly. Then next is there are certain techniques that are used. One is randomization. Randomization in experimental studies. Randomization means that you identify the treatments, 4 treatments A, B, C, D. Okay, and we want to test these 4 treatments in 4 groups of animals, labeled as group 1, group 2, group 3. And by following a random mechanism, you identify to which group of animals the first treatment will go by a lottery mechanism. Very simply known as randomization. Today, random number tables are available in all the scientific literature. You can make use of them. What is needed is you serially number or name the experimental units. Similarly, identify the treatments labeled as 1, 2, 3, and then make a matching randomly using the random number tables. So this way, in all the experimental studies, whether they are laboratory investigations of physical, chemical, or animal studies, or even clinical studies, you have to follow the randomization. There is another technique in clinical trials followed is known as the blinding. So blinding is a type of randomization technique in which the investigator who is doing the research investigation is not knowing which product he is giving to which subject. And hence the products are coded by the sponsor and supplied, and then again the allotment is made randomly, and the person who is taking the drug or the investigator who is distributing the medicines for test, both parties they do not know what they are taking, what individuals is taking, and hence this will avoid bias. Otherwise, the possibility is even there is an adverse remark. The way in which the clinical studies are being conducted, not now. Now they are recently checked, but earlier 25, 30 years back. So a lot of bias is being introduced in conducting the clinical studies with the result is always as expected only. That is not a good research. And later on, now globally, the regulations are prescribed, controls are prescribed for clinical studies, and hence today it is not possible to introduce any sort of bias into the product. And in drug research, what is the scope for bias? Yes, there is a lot of scope because it is a research, but it is a commercial element is involved. Some business element is involved, and hence it is a very possible human tendency to introduce bias. To avoid that, the only thing is that you have to make use of the randomization techniques. So use of placebo. One way placebo means without any drug. It looks similar and hence mixed up with the actual drug and they are given, and hence nobody can identify what is that placebo. So use of control group also come under the randomization only, and hence by comparing with the control, whatever effects or influence are there in the control, they are nullified for the drug treated one, and hence the correct assessment you can make it. Next slide, please. Next. 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 Next slide. Balan. Okay. Not moved, sir. Okay, okay. It is moved now. So the second principle is error should be minimum. Error should always be minimum. That means when you are doing the experiments, it is always possible to get some error. It is inevitable. So what is the purpose of doing experiment? Any experiment is to know the unknown parameter. There is an unknown parameter, true unknown parameter, which we don't know. To know that only, we will do certain experiment and estimate. So estimated value is taken as a measure of the unknown true parameter. And it is unknown, we made some assessment, and hence it is possible always to have some error. So the difference in the estimated value and true value is otherwise called error. And error, that type of error arises due to two types of reasons. One is due to chance factors, random causes, which will always be there in any investigation, which are beyond our control. And these are chance factors or chance forces. And the variability in the data due to such type of chance forces are known as the chance variability. For example, if you repeat the same experiment under identical condition, under same temperature of 40 degrees is used, same pH of 4.0 is used, everything same, conducted 6 times, measured 6 yields, and all the 6 yields will never be equal. So what is expected, though, because the experiments are done under identical condition, it should be the same, but never. The reason is because of the chance forces. The chance forces here are the minute changes in the parameters when you are conducting the experiment. Temperature, you say 40, but in one run it may be 40.05, in another it may be 40.1, 39.8 like that. And this little change in the temperature, you cannot identify, you cannot measure its effect individually, but collectively, many such things will influence the result, that is known as the chance variability. it will always be there. And because of the chance variability only, we are not certain to make conclusions, because uncertainty will prevail for the conclusions. And hence the second type of reason is for error is assignable reason. Assignable means there is a noticeable change in the factor. Temperature, in one run you kept 40 degrees, in another run you kept 50 degrees. So there is a lot of difference. Okay, 50 minus 4, 10 degrees, 10 unit difference, whose effect can be measured. Because of that real factor, the yield is different. Like this, you have two types of factors. And then the error due to assignable reasons, they can be controlled. They can be controlled. Our aim in research is always to keep the error minimum. To keep always the error minimum. And hence, to keep the error minimum, all the methods, techniques, procedures are to be standardized and used as per a defined SOP. So this is, again, these concepts we study in quality assurance. Okay, SOP. SOP is laboratory also, it is very important, and you need to use the methods and instruments as per a specified procedure only. That should be identified first and write down clearly. To repeat is supposed to get the same result. So all the manipulations are to be done in the uniform way, and hence SOP is very needed. So standardization is there, and validation. Instruments are to be validated. So for the most widely used, whatever is the lab, type of lab, microbiology, pharmacology, physiology, whatever it is. So you see, you agree that if I say balance is the most widely used analytical equipment. See, the balance is not of good quality. It's not working properly. There is no meaning in doing the research with that balance, and hence balance has to be validated regularly, almost daily, because balance is so frequently used. The requirement is that in the GLP guidelines, the requirement is that such machines, instruments like balance are to be validated daily, and should be certified by a competent person. Then only the researchers have to use it. The same is followed in industry, but in academics, how far this is followed is a questionable task. And so validation is at most important. All procedures, equipments, everything should be validated, and then only you will get the reproducible values. Means the accurate and precise values you will get. And then acquire necessary skill. This is again important, as acquire necessary skill. So that is the reason why the pharmacy programs are multidisciplinary course. So different variety of subjects will be there, and the student and faculty should get trained in the subject, not only theory but skill also. And here is a major concern. So this is acquire the necessary skill for doing research. So for example, when a faculty studied MPharm 10 years back, 15 years back, and those days the skills are different, nowadays the skills are different. And he certainly has to acquire again, get him retrained in this subject. For example, conducting a nano particle research, for example, okay, I don't think that the faculty who graduated 10, 15 years back, they are exposed to such type of experimentation, and hence necessarily they have to do again undergo training. This so happens that in the biological research, in the areas of pharmacology, a lot of skill is involved, and hence certainly the skills are to be updated frequently. Similarly, instrumentation skills are to be updated periodically. Then only we can do the meaningful research. Next. So certain, to some extent now, earlier we see that the error should be kept minimum. The second point, the error should be kept minimum. Now the fundamental question is, you ask why error should be kept minimum. Why should be minimum? What happens if the error is large? Yes. If the error is less minimum, they are known as the accurate values, accurate result. So all experiments design should give accurate estimates only. If error is more means we cannot find out the significant real differences between the estimates. Suppose, for example, two average values are there, two average values. Okay, the difference between the two averages is very less, but it is real. It is real, but very less. In such a case, if the design of experiment, if your way in which the experiment is conducted gives large error, then this large error will nullify the small real difference between the two averages. This you can easily understand if you remember the t-test of significance. So t-test statistic t is defined as x bar 1 minus x bar 2 whole divided by square root sigma by n. So this square root sigma by n is otherwise called the standard error, and hence the difference in the two averages is compared against the error. If the error is more, it will nullify the real small difference, and hence you will be getting the miscellaneous conclusion, erroneous conclusion. So error should always be less, and hence our experimental design should be such that you should get the always accurate results and precise. What do you mean by precise is when repeated under identical conditions several times, all the estimates should be the same. A measure of precision is otherwise called standard deviation, and hence uh, the standard deviation and standard error, which are measures of error, okay, they are related to the variability, and hence if the variability is kept less, the method is known as the more precise one. And now coming to this study design can be used to control and reduce the error, to reduce the, how it is in some designs inherently they may make use of the randomization principle, and with use of controls provision will be there, and adequate replication possibility is there, and hence we have to select a design which is having all the good qualities, like there should be a provision for replication, and there should be a provision for controls, and randomization principle should be applied, and the local control should be possible to apply, and crossover designs to nullify the inter-subject differences. So such design components will also contribute us to reduce the error. And hence, for example, crossover studies. So they are the preferred designs for all biological evaluations, either animal or clinical. And this is because in the animal and clinical studies, there will be a lot of variation in between the subjects. No two individuals or animals are identical, and hence the effects will be certainly different due to the inherent variability, inter-subject variability. To reduce its influence, which is a major part in the error, to remove its effect, now the best procedure is best technique followed is a crossover. Means just both the treatments should be tested in both the animals or in both the groups. So for example, in simple sense, take test and standard 2. And this week, first subject the group 1, give the group 1 test, group 2 standard. After some time again, reverse the treatment. The group which received test will now receive standard, vice versa. While doing so, if at all any effect of subject is there, it is equally applied on both. With the result, its effect is nullified. So real difference due to test and standard only we can achieve. Like this, for all biologicals, the best design you have to select is only the crossover design. Then why you crossover design for biologically? Just to reduce the error only. And why error should be reduced? To get the sensible comparisons and to make valid reasons. Similarly, in the case of other sciences, including biologicals also, the another important principle is, okay, is local control. Local control. So local control means it is an experimental design. So when the experimental units are very homogeneous, not sometimes, okay, very homogeneous, the experimental units are divided into groups in which the experimental units are more uniform. So for example, this concept is developed first in the agricultural science research. So where the fields, the fertility of this field is highly variable. Land near a stream or a river is more fertile than a land away from the river. And hence, the agriculturalists, when they want to study the investigation, all the crops are to be grown, all the varieties of crops are to be grown in an area nearer to the water canal or far away to the canal. That means the area can be divided into two segments, nearer to river and away to river. And then all the treatments should be tested in both. And hence, the variability due to fertility of the land is equally applied for both the varieties so that their influence is nullified and the real effect is found. That is known as the local control. In the same way, in the chemistry laboratory, the local control principle is well applied when you want to study two factors simultaneously on the reaction. So like many times in the chemical laboratory, the factors of importance are, say for example, temperature and pH. So one factor is considered as a block and another is considered as a real factor treatment. Suppose all pHs, 4 pHs are there. So at each pH is considered as a block. And if you have the 4 temperatures, all the 4 temperatures are tested at each pH, giving rise to a concept known as RBD. It is an application of the local control principle. So all these are just to reduce the error only. Now coming to the measurement of error is possible. You should be able to measure the error. So you have several statistical quantities like standard deviation, standard error, Fiducial limits. Okay, all these are error measures only. And then as the sample size increases, sample size designated as small n, if this is increased, normally the error gets reduced. So if you verify the statistical formulas in all the formulas of errors, the n value is in the denominator. If figure in the denominator, if you increase, the quantity will reduce. So error will become less. That does not mean you have to repeat a number of times, 100 times, 1000 times, not necessary. So sample size is very important. And even all the official books also for all the quality, even simple quality control tests, replication they identify. Not a single test is adequate. So for simple weight variation, test of tablets, capsule, the pharmacopoeia says start with 20 tablets. For disintegration, 6 tablets. For dissolution, it's a very complex test. So 3-stage test, 6 plus 6 plus 12, total 24 runs you have to do for making a final comparison. Like repetition is important, and in the same manner, in all our experimental investigation, in the scientific investigations confining to laboratory and other studies, so reasonably a small sample size is adequate, because doing a number of times, like 50 times, 100 times is waste of time and resources, not recommended. So, but whereas in several other studies like observation studies, okay, the pharmacy practice, clinical studies, in clinical studies also, is testing only in 2 or 3 or even 6 to 10 subjects is not adequate. In the clinical phase 1, phase 2, phase 3, the difference is only size is increased. In the initial, it is up to 30 only. In the next is up to 300, and in the final third phase, it is anywhere between 3,000 to 5,000. So the size is only increased. So in such studies, you can go beyond hundreds, etc. But all otherwise, for the laboratory investigation, better a small number is adequate. That number you have to estimate. Now, next slide, please. Next slide. So this is the same thing. Determination of the sample size. How to estimate? Yeah. So it is for determining the sample size, you require some data. What are the data is? Okay. One is the particulars data needed is accuracy or error that you can tolerate. How much error we can tolerate in our results. So there is no point in fixing the error at a higher level. Normally error should be at a minimum, and hence that you have to fix up, because the sample size required also majorly depends upon how much accuracy you need in the data you need. Suppose if you do not accept even any error should be practically zero, then size should be large, means the entire population you have to study. If you study the entire population, that is capital N, when small n is equal to capital N, that is otherwise means if the entire population is studied, then there will be no error. But it is not possible several times. So in all other cases, no, a suitable number has to be calculated based upon the accuracy needed and the level of significance, level of confidence or significance. So that is indicated as the probability of committing the type 2, type 1 error. That is, while making decision, possibility of 2 errors, type 1, type 2. Type 1 error means rejecting a true hypothesis. That type of error you have to fix up a level first, probability, that is known as alpha. Probability of committing the type 1 error is otherwise known as alpha. So you will find in the research papers and books, statistic books, alpha is taken as 0.05, and alpha, otherwise alpha can be taken as 0.01, means 5% level, 1% level. What does it mean is, the given statement is applicable in 95% times if you select alpha value as 0.05. The remaining 5 times the statement may be invalid. Okay, that means the estimated parameter, if the alpha is defined as 0.05, that means the estimated value will 95% times it is correct. The remaining 5 times it may go wrong. And hence this is otherwise known as, what is the confidence, with what confidence you are telling the statement, you are presenting this result. That should be indicated by prescribing the alpha value or level of significance. And then degree of variability. What is inherent variability in the data? In such type of experimentation, what variability will be there? So knowing these 3, you can easily estimate the sample size, which is in experimental investigation, the number of replications needed. I have the next slide, please. Next. Next. Okay. And here is, this is, the equation is derived from the equation given for fiducial limits. All of you might have studied in pharmacology. Okay, fiducial limits. These are the limits in which the unknown parameter will lie with a defined probability, like 95% confidence, 99% confidence. The same equation, if you can use it by rearranging, this will become like this. And hence, in this simple example given here, okay, this is an investigator wants to estimate the mean birth weight population. The standard deviation is given as known as 450, and the expected difference tolerated is given as the 30 grams. Then the sample size formula is n is equal to z square. What is the z? Is normal standard variate. Standard normal variate under the TD normal distribution z. That value in the statistics, the standard value at 5% level of confidence, it is 1.96 square, and the sigma square means it is the standard deviation. Standard deviation is given as 450, and then D, that is the difference allowed, difference which we can accept related to accuracy, that is given as 30. Substitute, and the result is 864. That means in this case, the sample size n is 864. Means if 864 subjects are verified for the birth weight, babies were verified for birth weight, then the purpose will be served. That means the mean estimated will be within the unknown parameter by a distance of about difference 30 grams. Okay, that is otherwise a sample is estimated based upon sample size, based upon three important things which you have to record. And hence, if you plan any experiment, you should first think about, okay, now what is the accuracy of the result? Accuracy here means that is the standard mean and sample mean, difference in the population and sample mean is given as 30 grams. In the same manner, depending upon experimentation, you have to first fix that. Then the confidence interval, you have to fix it. Normally at 5% only always. Or sometimes in biological experimentation, 1% is also taken, or the 5%. Now here considering the other things like, because 1% is more stringent than 5%, and hence the number of experimentations needed may be more if you select 1%. So by considering the other aspects like the resources required for test, as well as for the test procedures, etc., complexity, etc. So it may be either 1 or 5. Anything can be fixed. So like this, and similarly, if you want to compare the difference in the mean values, two samples will be there. In such a case, a simply modified formula, which is also just similar to the above. Like this, it is not exhaustive. In the statistics, there are a number of studies, cases. So you have to, if you go through that, and depending upon our situation and more appropriate one, the technique can be followed to estimate the samples. And the next one, please. So the next is this is already we discussed clearly defined objective and scope. So scope refers to the area over which the findings of this investigation can be applied, whether it is narrow or wide. So that also you have to define, because basing on that only, you will work out the treatments, okay, and groups to be included in the study, etc. That is otherwise called scope. Suppose you are evaluating the efficacy of a drug, okay, in an animal group or a human groups, etc. So age is one important factor. Age is one. So if you confine your study to adults only, for example, whatever result you get, it is applicable to adults. Instead, that means the scope is narrow. Instead, if you want to broaden the application, because the same drug may be used for children, infants, or geriatrics, etc., in which case, to enlarge the scope, you define first to evaluate the efficacy of this in these age groups, starting from, say, infants, children, like that you define. In such a case, the experiment should be designed in such a way, including all age groups of people in the study. Then the result is applicable to all the age groups. That's why I think scope. So initially, you have to think about the scope, possible area of application. And usually, the scope should be broad. Next, literature survey. Because first, initially itself, added the word literature survey. So there are wonderful ways of conducting the research survey. The sources of information are, the primary sources are the journals. They are always the best source you can relay, because original authors, original details will be there in the journal only. But whereas for quick search, initially you can go to secondary sources like abstracts. There's so many abstracting agencies have come, and continuously they will be supplying. But the difficulty is sometimes they miss the details. This is because the abstracting services, sometimes they use the abstract made by the author. Many times the reviewers, they themselves will condense the article and make their own way abstract, and that is included as the abstracting source, abstracts. If you read that, you will get information, quick information, but details are missing, and there is no guarantee that they really reflect what is there in the article. And hence the best way is for quick reference. Initially, you have to screen all the abstracts only, going through various abstracting journals, then identify, then go to the original article wherein you will find every detail. That is more important. Every detail in the sense theoretical one, literature detail as well as practical details you will get Those we have to understand clearly. From that understanding only, you can find out the areas where we can further, we have further scope to investigate. And next is tertiary source. This also should not look it down, but it is standard textbook. Standard textbook. Now, reading of the standard textbook in the academics is going down year after year. Okay, so nowadays both the students as well as the faculty are confining to only small, okay, books derived from other sources, etc. But original books, if you are interested more in research, certainly for understanding the subject in a better way and learning, there are standard books. Okay, and for example, in the area of organic chemistry, everyone know that the medicinal chemistry, Budget's medicinal chemistry, the very voluminous books are there, voluminous books. But as a student, as a teacher, it may be difficult to study that for the regular class and teaching. But if the teacher is interested in conducting a research, so certainly the teacher and student can sit and read more thoroughly and improve their knowledge and understanding of the subject. In that way, the reading the standard known as the tertiary sources is very, very important for research. So this is otherwise based upon the literature only, the objectives will be drawn. Next. Okay. 8. So it is methodologies and protocols are to be described in detail. It's very, very important. Methodologies and protocols. So methodologies means they refer to the details of the experiments to be done. Whereas protocol means all details right from the objective literature till end. Okay, protocol. What are the details are to be given? In a way, both are same, but protocol is accessed. Methodology is a part in the protocol. And then, so the purpose of this is to permit any other person with some knowledge and skill in that subject to repeat and get the same result. That is very important. So when once you write an article based upon your research article, that article should also give all the details. The purpose is, suppose if some other investigator go through that article, and if you want to repeat it, when he repeat, he should get the same value. When he repeat, he should also get the same value. That is the purpose. And hence, in the methodologies, every detail you have to give, full length. So he refers some standard articles in national, international standard journals. Every detail, minute detail they ask, and you have to give it. Okay, for the validation of the work you have done, the way in which you have done, this is very important. So here that is, as far as possible, official and other standard methods are to be used, but official methods may not be available for all aspects. Then in which case, you have to develop, whenever a method is developed by the investigator, it should be validated appropriately. Validated especially for accuracy and repeatability, without which it is not accepted. So all the validations, okay, now a lot of information is given in ICH guidelines for analytical method validation, WHO guidelines, several other guidelines are there, and the GLP has given guidelines. So following them, when you use a method, newly developed method, because it's not there earlier, so you have to be very thorough in validating that, and then only you should confirm yourself that it is accurate to the level needed, and also it is reproduced. Suppose if it is not developed by you completely, but is taken from source. Now identify that source. Select only the good sources. Now there is a lot of variability in the standard of the journals in which these research articles are published, and hence you cannot equate all, and hence judge by the journal quality, article quality, and reputation of the group which are doing the work, presenting this work. And then even then, if you follow that, give a reference and then you check its suitability. Means again you have to evaluate for its applicability, or essential parameters you have to revalidate before using in our study. So all methods are to be validated for reproducibility and accuracy. Okay. Then SOPs are to be developed and are to be followed for all methodologies, and all studies and details or other materials, instruments, everything should be included. So all these specifications and other details of not only drugs, excipient materials, reagents, instruments, animals, microorganisms used, their specifications, everything should be recorded and indicated in the protocol, and will be assessed by the committee. And then intervention means drugs used, everything should be spelled out clearly in the protocol. Then statistical methods and the software that you are going to use it, because several times nowadays nowadays several statistical techniques to use it. It's not by manual usage. So software has been developed, and hence you have to mention which software. Pharmacokinetic analysis. There are so many software. Even statistical software are there. And all those things you have to identify and prescribe. May I have the next slide, please? Next slide. Next slide. Next slide. Jaybalan. Dr. Jaybalan. Okay. Okay. Good. So protocols are to be described in detail, and the protocols are to be approved by ethical committees before commencing the research. Whatever is the research, that is very, very important. So that means what is protocol we have seen every detail, but that should be approved by some committee known as ethical committee. So different for each study, for clinical study, the ethical committee is known as IEC, Institutional Ethical Committee. For animal studies, the committee is known as IAEC, okay, Institutional Animal Ethics Committee. And for laboratory studies, and the protocols are to be approved by a research committee or a college level. These are college level, because there no other body is there to give guidelines for approvals for laboratory studies. But laboratory study protocol should also be drafted quickly and discussed in the meeting of the committee. And what is that committee? It is known as a research committee or college committee. Okay, in the revised PCA syllabus for BPharm and MPharm, there is general provisions. The PCA has recommended the constitution of a college committee for taking up the decisions regarding the teaching and research activities. And hence, such college committees at the college level will thoroughly check. And why the checking is, the investigator will certainly take care, but even then, sometimes one or other aspect may be ignored by the investigator, either purposeful or willingly or unwillingly, knowingly or unknowingly. In such a case, the expert committees sitting in these committees will review and view it in different ways. How far it is legal, how far it is ethical, how far it is safe, is it okay? And they verify all aspects, and finally they give the approval. May I have the next slide, please? Next slide, please. Okay. Now, principle 10. That means documentation. So today, documentation is utmost important, not only in industry, but everywhere in the academics also. Documentation is utmost important. And the type of documentation needed for research proposal is, so project proposal is one. Initially, you have to prepare and submit a project proposal that should be preserved properly, and protocols, detailed protocols, which are to be, both these things should be approved by the committees said earlier, and they should be preserved, and minutes of the ethical committee meetings and all the meetings held for approval, sometimes midterm review and final review, all these things should be stored properly, preserved properly. and all letters of approval and consent letters from participants in clinical studies. So because clinical studies normally involve humans only, so they should willingly give acceptance. So the consent letter in the prescribed pharma has to be taken from each subject and should be preserved well. And reports of project work done with all details and results should be preserved. And any other document that the sponsor needs. So this is because for sponsored research, it is two types: government sponsored research and industry sponsored research. So in which case, either the governmental bodies or industrial bodies, they may insist some compulsory document for verification. So all such documents are to be prepared and carefully and to be preserved with the principal investigator or project director. Well, the next slide, please. Next. Okay. This is the now in the research. Statistical testing is at most important because, as I said, the result of any investigation is influenced by the chance forces and the real forces. We are interested in finding the real effects, real factors and its influence. But simultaneously, there will be chance forces which will be disturbing always. And hence, finally, there will be a lot of confusion whether the observed result can be taken as the effect of treatment or not. And hence, the best way is that in such a case, using statistical technique, the uncertainty in the results can be avoided. Uncertainty. And hence, now we are more concerned with the experimental studies and observation studies in which normally a lot of data is compared, and the one important, apart from other techniques, there are so many other techniques of statistics are useful, but among all, the analysis variance, otherwise known as ANOVA, is the most useful technique. And what is this is, analysis variance is a statistical technique to find out the significant differences among several averages. If you have only two averages, a simple t-test can be used depending upon the sample size. Normally in scientific work, small sample only, so you can make use of the t-test. If the sample is large, you can make a z-test. But if the averages are more, suppose if you are studying the effect of 4 temperatures on the yield, so corresponding to each temperature, you will have one average, 4 averages. Then how to compare it is only possible by ANOVA, analysis of variance. The name has come because the analysis of variance, that means variance present in the data, whole set of data is analyzed and a decision is taken about the averages. And that's why it is called analysis of variance. So the term indicates variance, but it is useful to find out the significant difference of the average only. And how it is the basic principle involved is that in any given set of data, okay, for example, you consider the case of testing 4 temperatures and the yield of a chemical reaction. Each temperature replicated 4 times, 4 into 4, 16 observations will be there, and this is total data. And the variance present in the 16 observations is due to 2 reasons. One is due to chance forces, which are beyond our control. The variance due to chance forces, otherwise called a chance variance or residual variance, and the variance that arises due to the real factor temperature is known as the variance based upon the variance real difference, real variance. And hence the total variance present is divided into 2 components. One is the chance variance. How it is estimated is, so variation within the sample, within each sample, take what is one sample of 4 observations that corresponds to treatment 1, T1 is considered a sample, and estimate the variance in that sample from the corresponding average. Similarly, take the second sample, estimate the variance from the corresponding average, and then sum up. This is the total residual variance. this total residual variance represent the variance due to chance force only. It will always be there. We cannot minimize or reduce. Okay. You cannot avoid. Of course, we can minimize by resorting to various things which we discussed earlier. Okay. Then it is independent of any difference between the sample means. It is nothing to do. This residual variance is nothing to do with the sample difference. And hence, what is the other one? The other component is the other variance based upon the sample means. Now, write down the sample means. What is the mean of the first treatment, second treatment, third, like 4 means. Consider it as a series and estimate variance. This variance is due to the differences in the sample or difference in the factor. This is real. And hence the variance is divided into 2 components. Both are variance quantities only. Hence the difference between these two is tested by an F ratio test. F ratio test is a test of significance for variance quantities, whether the 2 variances given are same or different. So a significant F ratio here indicate that the differences in the average are really different, significantly different, and hence all analysis is made based upon the variance, but decision is taken over the average. That is possible because the 2 variances are calculated in 2 different ways. The chance variation is due to only chance factors, whereas the second component, which is based upon the averages, it includes both the effect of average, means treatment, as well as the chance, and hence comparing these two will enable us to make a decision about the average. Next slide. Okay, this is our next. Okay. before this. Okay. So here is one simple data is given. Okay. This is all the way in which you can do the analysis. And for back previous one, sir, previous one, back one. Okay. Now, okay, this is good. Okay, here it is. You see that there are A, B, C, D, E. That means 5 treatments. I am using the word treatment. What do you mean by the word treatment? Is treatment describes the conditions under which the experiment is done. Suppose the A is done at 40 degrees, it is one treatment. B is something different, 50 degrees, another treatment like that. So 5 treatments are there. Each treatment is replicated 5 times, like replicate 1, 2, 3, 4. This is the data. What is the total data available is 5 into 4, 20 observations are there, n is equal to 20. And then in the statistics, you find a sequence of calculations in the form of a table. Next chart. Next slide, please. Next. Okay. Now this is what way which dividing is. Next, previous one, sir. Previous one. Okay. Now here you see that it is a total variance divided into 2 groups. One is variance between the treatments related to factors, and variance within the sample means error. So they divide into 2 components, and finally it is tested by an F ratio test. Next slide please. Next. Okay. Next slide please. Okay. So this is the ANOVA gives you whether the 4 averages are different or not. That's all. So in the said case, there are 5 averages, and 5 averages are significantly differing. Next question is, which of the 2 are different? Which of the 2 are different? Which of the 2 are not? For that, in the statistics, we will provide you a quantity called least significant difference, LSD, or critical difference, CD. This is defined by the simple equation. If you substitute the same, the values calculated earlier will be useful to you based upon the error mean sum of squares and sample size, and such that the value is 3.81. and hence this is the least difference which we can consider significant. Okay, and hence you can combine as pairs. There are 5 A, B, C, D, E, and hence you consider first the averages. A average is 6, B average is 10.5, and hence if you take A and B, what is the difference? The difference is 10.5 minus 6, 4.5, and 4.5 exceeds 3.81, and hence this is significant. So AB is significant. Difference between AB is significant. Like that, among others, so you can see that, so you can easily say, initially first statement is that all the 5 averages are different. Next statement is that which of the 2 is significant, which of the 2 is not significant. For that, the best way is to calculate LSD or CD, and hence this is useful to make such conclusions. Next one, please. Okay. And selection of study designs. So statistical experimental designs provide you standard designs which are developed theoretically as well as practically. They are well established, and these are listed here. Types of designs for experimental studies. Experimental studies may involve animal or clinical or laboratory anytime. This design is useful for all laboratory designs, laboratory studies, animal studies, clinical studies. So CRD, RBD, LSD, these are very famous, and then factorial designs. And now coming to the next one. Next slide, please. Next, okay. This is what is known as completely randomized design, CRD. As the term indicate, it is completely randomized. It means the allocation of treatments to experimental units is made completely at random by a random mechanism. So this is only one factor can be studied, not 2, only one. So if you want to study the effect of temperature on yield, you can do it. If you want to study the effect of binder on the dissolution rate, you can do it. Like that, it is possible to evaluate only one factor at a time. But in pharmacy, you come across several times such cases, like examples are given to evaluate the effect of 4 binders on the dissolution rate of tablets. So here the factor itself is binder. But I say 4 binders mean they can be considered the 4 levels of the binder. Okay. And sometimes the binder concentration. So 4 concentrations of the same binder. Like that, it can be interpreted. And to evaluate the effects of 4 temperatures on the yield like that, only one will be there, one factor, but that factor has to be studied at different levels, or the same thing may be replicated. So here advantage is any number of factors can be studied, and each factor can be replicated any number of times. That means flexibility for the designer, and hence it is a simple laboratory experiments, investigations of physical, chemical, all are being done by this method only. And the total variation is due to chance factors here, and one is chance, the other is the variation due to the factor involved. There is only one factor, and hence the other component is variation due to that factor, either temperature or like that. So here we are not taking care any control over the inherent variability or biological variability, and hence this design is not suitable for biological experimentations. So it is only for physical and chemical studies. Simple flexibility is there. Okay, and because suppose if you are testing the effect of 4 binders on the dissolution rate of paracetamol. Okay, so each binder has to be repeated 4 times. So now total number of batches of paracetamol tablets required is 4 into 4, 16. So paracetamol is a simple compound, of uniform quality, can be made easily available, and hence there is no variability in the experimental units. So that you divide into 16 parts, label it as 1, 2, 3. Take first binder acacia and randomly pick up 4 numbers and allot this first binder to those 4 lots of paracetamol and carry out your experiment. So like this, the execution of this is also very simple. Okay, and next one, next design. Okay, this is next one. Randomized block design, RBD. Randomized block design, RBD. So here the experimental units are divided into groups. That means where there is a heterogeneity in the experimental units. Sometimes, as I said earlier, agricultural experiments is much developed. But in all other areas also, wherever variability is there in the experimental units like subjects, animals, lots of materials, etc. Okay, in which case it's not good to check all it randomly in the experimental units. If you do that, it creates a lot of error because difference are there in the unit itself, and then error will increase. So in such a case, the best way is applying the principle of local control. Divide the experimental units into more homogeneous groups. In the case of biological, ultimate division is each subject is taken as a group as more homogeneous. Or otherwise, sometimes the age-wise, the factor of variability taken as age. So all the children below 12 years considered as one age, and 12 and say for example 40, another 40 to 60, above 60, like that. Based upon the age, you can divide the subjects into groups, and in each group all the 4 treatment drug products are to be tested. That is again come under the randomized block design. So here the advantage is based on the heterogeneity, the units are divided, and after division they are known as a block, and in each block you have to test all the products such that whatever variability is there due to block is exposed to all the treatments and hence is nullified. So the result is very sensitive. So to reduce the error, this type of procedure is followed in several studies. And many times in pharmacy, when you want to evaluate the effect of 2 factors, like temperature and pH, for example, okay, and one factor is taken as corresponding to block, another factor is as a treatment, and with the result, suppose 2, the total variance present is now due to 3 reasons. One is chance variability, it is always there. The other is variability due to block. Suppose if you coincide block with pH, variability due to pH, and treatment temperature, variability due to temperature. Like that, the total variability is divided here into 3 components. Whereas earlier in the completely randomized design, the total variability is divided only into 2 components because there is only one factor under investigation. And then in the same way, the analysis of variance is done. ANOVA is useful, but ANOVA here is called 2-way analysis, whereas in CRD, it is 1-way analysis. Based upon the one factor only, the values are separated and estimated. Here it is in 2-way. In 2 directions, the data are to be classified, and hence in the same procedures are used to estimate first the total variability, next variability due to block, variability due to treatment, and residual variance. Then at the end, 2 F ratio tests are to be done, one for block effects, another for treatment effects. Basing upon the significance of F ratio, 2 decisions are taken with regard to the effects of block and the treatment. And hence, this is a most widely used design in pharmaceutical research of all types, including biological research, etc. Next slide. So here it is again, the same thing is written, and examples like to evaluate the effect of pH and aeration rate on the yield of an antibiotic by fermentation method, bioequivalence studies, and pharmacological studies. This is most widely used. And to evaluate the effect of, say for example, in formulation, disintegrant and binders on the dissolution rate simultaneously. So several times, okay, both are included. So you can select some 4 different disintegrants and 3 binders, and then plan your experiment as per an RBD and analyze to find out the significance of the disintegrant effect and binder effect like that. So an advantage is experiment error is much reduced. Experimental error is much reduced, and hence you can make out very sensible comparisons. That is the reason it is most widely used. So next slide please. Next slide. This is a Latin square. So this is here in the same way, the experimental units are divided in two directions based upon two factors of variability. Two factors of variability, they are divided into, and hence two factors are taken for division. So a third factor is possible to study its effect. For example, in a chemistry laboratory, we know that normally pH and temperature are the two factors. In addition, a third factor may also be interested, like a catalyst. Either type of catalyst or concentration of catalyst may be important, in which case there will be three factors. So this study is useful when we want to study three factors at a time on the phenomena. And many times three factors are involved. So in physical, chemical, and such other and biological studies also done according to LSD only. A good example of LSD application is the bioequivalence testing, comparison of products. Okay, and hence here it is in this small table here. It is a model of bioequivalence testing. So subjects 4, 1, 2, 3, 4, and M1, M2, M3, M4. That is a time period. So in a bioequivalence study extends over a longer period of time, because all products are to be tested in each and every subject, and a washout period of suitable length has to be given. With the result, it extends over a longer time. In such a case, the time trend will be influencing the result, especially ADME character. And hence, here the three factors involved are A, B, C, D are the products for the bioavailability of which is to be evaluated. So the three factors here in the study, the bioequivalence study, is one is the subject variability, the other is time trend effect, the third is a product in which we are more interested. And hence three factors are arranged, and for allotment you have to follow a Latin square design. This is a square having equal number of rows and equal number of columns. It is a 4 into 4. It is a 4 into 4 Latin square. With the result, you have the 4 into 4, 16 cells. And in the cells, the treatments are allotted. So in the given example for bioequivalence, the subject was 1, 2, 3. So each, say for example, each row is representing one subject, second subject like that. Each column is representing a time period M1, M2, M3 like that. In between you have the ABCD products. So the products are allotted to the subjects and time periods in such a way that each product appears only once in each row and each column. Okay, in each row and each column. That means here it satisfies the rule that all the subjects are receiving all the 4 products, and all the 4 products are being tested in all the 4 time periods, and hence their effects are nullified, and the real effect due to the product can be observed because the error got much reduced. So it is a further extension of RBD, LSD, which is very much useful to reduce the error, especially in biological evaluations like bioequivalence. FDA bioequivalence recommended this only. And then in the other studies also, as I said, for chemical, number of times the factors involved maybe 3, in which case it is more useful. And here if we look at the diagram given subjects, the replication appears to be 4 only, because 4 subjects. And so if 4 is not at all a suitable replication for a bioequivalent test, and hence if you want to improve the equivalence, instead of 1, you can take 10 subjects in each. So instead of 1, 10, then another 10, like that, it increases. Okay, 4 will become 40. So 10 times in each group we can increase. But the rule is that all the products should be tested in each and every group or subject. So the main advantage is that, and how the data is analyzed. The total variance, next one. Next slide. Total variance is due to 4 factors here. total variance is due to 4 factors. One is due to chance, and the other is due to the product. Third one is due to the subject, intersubject. Fourth one is due to time trend. And in the same manner, at the end, the 3 variances due to product, subject, and time trend, each should be compared against chance by an F ratio. So 3 F ratio tests are needed to make 3 decisions. That is the advantage of lattice square design. And so far we considered the CRD, RBD, and LSD. These are wonderful designs, but in pharmacy several times these are standard designs. But the disadvantage of those designs is they are known as simple designs. Means that you can find out the main effects of factors, either 1, 2, or 3. But whenever 2 or more factors are involved in any study, in addition to the main effects, they also produce a combined effect. A and B together they produce a combined effect. It is known as interaction effect, and that interaction effect is not possible to test in the earlier designs. And hence, the other experiments which are known as the factorial experiments. These are the experiments which can measure not only the main effects but also the interaction effects. They are known as the factorial effects. So it is possible to measure both main effects due to A, main effect due to B, and interaction effect, combined effect of A and B, like that. And these are, so in this, it is a factorial study, and you have 2 factors, and each factor to be studied at minimum 2 levels. So in the designs, DOE, okay, design of experiments, to evaluate the effect of any factor, that factor has to be studied minimum at 2 levels. Okay, minimum at 2 levels. And hence here it is 2 levels each factor. Then if we have 2 factors, it is known as the 2 to the power of 2, 2 to the power of 2 factorial design. If you have, for example, say 3 factors, it is known as 2 to the power of 3. Okay, but factors may be different, 2 factors, 3 factors, but each level should be always the 2 only minimum. You can evaluate the effect, and hence these are known as the 2 to the power of 2, 2 to the power of 3. So how to execute this work is, first you have to select 2 levels for each factor. 2 factors A and B. For each one you have to select 2 levels again. So to find the effect of any factor, that factor has to be studied at 2 levels. So those 2 levels you have to first fix up. Lower level, higher level. With the result, the selected combinations needed for comparison here can be derived by expanding the simple mathematical static known as 2 to the power of 2. So 2 to the power of 2 is equal to 4, and hence in this experimental design, there will be 4 treatments comprising the selected combinations of the levels of the 2 factors, okay, A and B. And hence, there is a notation which we can easily implement it without without again going back to a statistician. And this is to expand it and write down the treatments. Simply first write down 1, okay, then A, next B, next AB, 4 treatments. So how to interpret is, when a letter is absent, means that is at the first level. When a letter is present, that is the corresponding factor is at the second level. So in the treatment 1, both A and B are absent, and hence both are at the lower level. In the treatment A is present, and hence A at higher level, B is at the lower level. In the B, the same way, B is at higher level, A is at lower level. In the AB, both are present, means both are at the higher level. And hence the design is like this. You identify the factors, fix 2 levels, reasonably having a difference to elicit the effect. Okay, that depending upon the phenomena, depending upon the nature of the study, etc., or character of the parameter evaluating, etc., can fix selected 2 levels. And then Then follow the simple rule. This is so 4 treatments you will get, and then execute these 4 treatments. You test as per either a CRD or an RBD or an LSD. So the testing is again done as per the standard design only. But here the purpose of factorial design is the treatments, selected combinations you can identify with this concept. And hence, when once you write down 4 treatments like 1A, B, AB, then you select depending upon the character of the experimental unit. Sometimes you can select RBD, sometimes LSD also needed, and hence carry out the investigation. Okay. So there generally we discuss the treatment. What is the treatment? Is these 4 treatments. And hence, may I have the next slide, please? Next slide. Next. Okay. Now this is here I have given to better understand this concept. So this is a simple case in pharmaceutics. Effect of beta CD, beta cyclodextrin, which has several applications in pharmacy, formulation. Poloxamer is also a non-ionic surfactant, and PVP, a well-known polymer, polyvinyl pyrrolidine, on the solubility of pioglitazone. This is a poorly soluble drug, pioglitazone. We want to improve the solubility using these 3 type of ingredients, and 2 to the power of 3, because 3 factors are there. What are the 3 factors? The 3 factors are A, factor A, beta CD, B, poloxamer, C, PVP. Now the 2 levels are needed for each, and hence for beta CD, 0 and 5 millimoles. It is solubilizing agent. Poloxamer similarly, the 0 and 2%, and PVP also 0 and 2%. Okay, like this. So in the case of beta CD, its strength is normally expressed as moles only, and hence that is by 5 millimole. Whereas in other cases, generally percentage only. So this is so how many? 2 to the power of 3. 2 to the power of 3. If you expand, there will be 8 treatments required. Earlier it is 4 only, now 8. Now in the same manner, how to develop the treatments here? First you write down 1, then you write down A, B. When 2 are entered, product of these 2, A, B, then enter the third factor C, then multiply C with the earlier one, C, then AC, BC, ABC. So all 8 treatments are written, and then execute the work. Execute the work means what is treatment A? 1 means all are at lower level. All are at lower level means what is the lower level? 0, 0, 0 means it is pure water only. It is only pure water. So determine the solubility in water. Because inherently we want to find out the effect of these on solubility. So first initially what is the solubility in water? Then you find whether addition of these materials will improve or not. So what about individual effects and combined effects together also, whether improving or not. So this is each treatment should be replicated 4 times or 6 times like that. As you like, you can fix it. Then these are the values of solubility. So mean is there, SD is there. Now this is simply giving the force increase, okay, and force increase the treatment. Now you know why you have to perform. Next slide please. Next slide please. Okay, this is all experimental designs in which more than 2 treatments are involved. The best way of analysis statistically is by analysis of variance only. So this is the way in which earlier we considered. The first step here is to find out whether the treatment effects are significant or not. So the probability P value is less than 0.01 means the treatments are highly significant. Means all the treatments are significantly different in their solubility character. That is a decision at the stage. Next comes, it is not a factorial means you have to find out not in total, no, individually whether factor A, B, C are combined, all the 8 that you have written, whether they are means main factors as well as combined factors are significant or not. So for that, an extension of ANOVA is made, and also variance. That method is known as the Yates method, and wherein the treatment totals are calculated, manipulated. Finally, the total variance observed there is now divided into, this technique is useful to divide the total variance into 8 groups, plus 8 groups means all 8 treatments plus chance, and hence this is the way. Okay, this is the way the analysis. So due to 1, what is due to B, due to C, etc. What is the variance that is possible? So earlier in all other CRD, total variance is divided into 2 only. In RBD, total variance is divided into 3. In LSD, total variance is divided into 4 components due to 3 factors and 1 chance. But whereas in this 2 to the power of 3 factorial design, the total variability at the end is divided into 8 plus 1, 9 components. And these first 8 components are to be tested against the chance individually by applying a series of F tests, and you can make a decision. Next slide, please. Next slide, please. Okay. This is what is done. Okay. Okay. This is what is done. A, B, A, B. So A means effect of A, B individual effect. Like that, AB means combined effect, and combined effect ABC means all the 3 like that. Okay. Now result indicate that the significance, you see, all effects are significant except that BC, except BC. So BC, the p value is greater than 0.05 means not significant. So what is BC? BC indicate the combined effect of the B and C. B and C means PVP is C, B is poloxamine. So surfactant PVP together may not be having significant effect, but whereas in the other cases the combined effects are significant when beta CD is included. And hence, like this, you can, the advantage of this type of factorial design is you can find out simultaneously the main effects as well as the combined effects. And hence, whenever possible, the experiments are to be organized in the form of a factorial design only, which is very much useful in formulation development and the modern optimization techniques as well as the QBD approach. Inherently, they are based upon the optimization upon these factorial designs only. And okay, so many software have been developed for applying these techniques of optimization and QBD, which are essentially based upon this factorial design only. Next slide, please. Okay, now coming to clinical studies. So clinical studies. So here, when coming to clinical studies, mean essentially studies carried out in human subjects. They are experimental and observation. So experimental means what we have seen earlier. The same thing is applicable whether it is done in a clinic, human subject, or animal, or in laboratory. The same concepts are applicable. And observational means something different. Okay, that is, here it is observational means it is only sometimes, many times, no intervention. Drug is not included, but effects are observed. While the patient is undergoing treatment, the symptoms are studied, the condition is continuously followed, and hence it is observed. Okay, and sometimes intervention drug is given once and then followed, observing the qualities only. Okay, no measurements are made several times. So like observation studies are most widely used in clinical practice and pharmacy practice. And coming to the experimental studies, an intervention is done and the subjects are observed for its effect. That means drug is given and the subject is observed. That is experimental. And include a control group normally to check the variation, intersubject variation is one way of using. Okay, a group without drug. So whenever we want to establish the efficacy of a treatment, okay, it is always necessary that one group of subjects should not be given any treatment and observe. So one group is treated, another group is not treated, and observe the effect or the mortality, whatever it is, and then compare these two groups and compare. And chi-square test is very much useful in such comparisons to compare the expected effect in the drug treated group and expected effect in the control group. By comparing these two, a significant difference only proves that the treatment is effective. Like this, the controls are very much needed, and especially in biological research, and include a control, and subjects are randomly assigned to groups, and clinical trials are also experimental studies. Next slide, please. Clinical studies are also experimental. They're a type of experimental only. Next. Okay. Types of designs. No, earlier one. Types of designs. So for experimental investigation, especially in the clinical lab, clinical studies, so designs are known as the parallel designs. So slightly different words are used to designate. Parallel means two groups are given two treatments, interventions concurrently, and effects are followed. So one group is, and that means two treatments are given to two groups separately. Each group is followed, and hence it is good, but it has a disadvantage. It has disadvantage means the subject variability is not nullified, because one treatment is tested in one group, another treatment is tested in another group, but the subjects may have their own effects, and hence care is not taken, and hence these parallel designs are not much used in the clinical investigations. Then in the paired design, this is again very much used. Paired design. Paired design means both the treatments are tested in each subject, in each pair. Okay, that means first a measurement is made in a patient or an animal, that is blank at 0 time before giving the drug. A measurement is made, parameter is measured, and then the drug is given and allowed some time to interact, and then the response is measured. Okay, response. That means the two measurements are in the same subject. Two measurements are in the same subject, and hence it is called a paired data. Paired data. The two observations, that means before treatment and after treatment, both are measured in the same subject, so it is called a paired data. And several times to evaluate the effect of drug, this is the best design, either animals or humans, etc. So before giving a drug, normally at 0 time you take a sample and process it and evaluate the parameter. Given the drug, and afterwards again sample is taken and processed. The difference is attributed to the effect of drug. Okay. And hence there are paired data, paired designs are used, and here again the effects are statistically compared by t-test of significance applicable to the paired data. And so here in this case only one group is used. After measuring the baseline 0, the dose of drug is given and then again measured. The difference is taken as an effect. Next, simple design pair, but much widely used, paired data. Next design. Next is, okay, crossover design. We've seen already several times crossover. Okay, the definition only given here. The purpose is inter-subject variation, time effects, and carryover effects are all nullified. And this is the best design for biological evaluations as well as clinical evaluations, so bioequivalence, etc. Next slide, please. Next. This is observational. So these are, so far it is all experimental only. Now it is called observational. So subjects are just observed for the parameter to be measured. That's all. Object observed and parameters are measured. No intervention is given normally in a hospital. You have to go on observing the conditions, and descriptive studies. This is observational. Another term is there, descriptive studies. Some are called descriptive in the sense that to describe the characteristics of the study, characteristics of the study subjects without intervention and hypothesis testing. So description is here. So just to observe the patient. In the case of a disease, newly has come. Okay, nobody knows what is it, but sometimes a doctor or a pharmacist come across and study the characters of this patient. So describe it technically. in the scientific language, and that is known as a descriptive study. To describe the characteristics of a disease. Say, for example, now we are all in the mid of COVID-19 pandemic. So just 6 months old only, and no one exactly knows what is this disease, what are the symptoms. So somewhere first the doctor who sees this, and he goes on describing the character only, characters of the patient, describe it. So that is called the descriptive study. Okay, next one, next one, next one, next one. Okay, and types of descriptive studies. Case report. This is case report is a description of an individual subject, a single patient, individual subject who has encountered with a rare disease. That is called a case report. And case report is the minimum publishable article in medical literature. So the first report or a disease which is not known, a rare disease, and hence just they observe the patient and the characteristics are described on a single patient. It is known as a case report. It forms the first report for any disease. Whereas case series, suppose the same, if extended to a group of patients, a group of people, it is known as a case series. A number of people suffering with the same disease, the conditions are described, the characteristics of the patients, and okay. And study of a random sample, a different type is known as the cross-sectional study. All these are almost same. Cross-sectional. So in the cross-sectional study, now you will get a sample of patient. A random sample of patients are taken and their characters are observed, a random sample, okay? And a series of samples can be studied to find out like the prevalence of the disease, risk factors of the disease. These are the important characters, risk. So different groups are observed for different risk factors, and a study of the description of the patients is done and reported. Such are known as the descriptive studies. So in the descriptive studies, case report forms the minimum first case, and then case series, a group of patients who are suffering with the same symptom disease, they are described. Alternatively, study of a random sample. Investigator will select a random sample. Random sample in this town, another random sample in another town. Like that in another country, random sample is taken, and then the symptoms and characters of the same disease is studied, and they want to try to correlate it with various factors. Sometimes risk factors, sometimes differences, race differences, gender differences, like age differences. So some assigned and inferences can be made. Such a study is known as the cross-sectional study. Next one. Next. So case control study. This is again an observation study only, case control. A group of patients with a disease is compared with a group of people without disease. This is a comparison. And normal patients are subjects. Okay. So when the disease, a group of people are kept as control, and then another group is the patients, and then both are studied for the observations made and compared. The result is compared to find out the characteristics of the suffering group, like that. And both groups are followed backward in time to determine the association with the risk factor. So this is backward, means retrospective. So retrospective means hospital records are to be verified, and they examine. This is to find out possible risk factors mainly, and the efficacy of the treatments also. Suppose if that time in the past, in the backward time, retrospective time, the certain prescription may be given and treatment may be given, and result may be recorded, and observing all those, you can assess the treatments also. And such type of studies require a retrospective study. Okay. And hence it is like a retrospective validation. What has already completed, verified through records, and that data in a control group, in a group of patients, is compared to find out useful information regarding the disease character. So this is again a case control study. A cohort study. This is also much similar cohort, but here it is a group of patients are followed forward into the time. Forward means progressive. Okay. Is followed along with a group. A group of patients or subjects is otherwise known as a cohort. Like they move together, like a group of soldiers in military, it is called a cohort. From there only it is derived cohort study. So a group of patients are identified in a hospital suffering with the disease, and then they are followed forward in time, and hence it is progressive or prospective study, and symptoms are recorded. Again, a description only. It is all descriptive study only. So several times like this, in observational studies, they come under the descriptive category only. Next. Okay. And then the principle 13. This so far we have seen about standard designs for experimental and clinical or observation studies, etc. And then next comes the principle 13. That means report writing. Report writing. This is also known as the scientific paper writing or report writing. So whatever research you do, it should be written, documented properly in the form of a report. That is otherwise report writing, which is just very much useful, scientific writing. So we should develop the art of scientific writing. That is very, very important for all the researchers. Okay, and the report should be in English, or if approved by the concerned bodies in a local language, but English is widely accepted, and then it should be very clear, simple English, need not very classical English, simple English, but it should give the clear ideas about it. And what is that written is the components always should have a title. The title should be very appropriate to the project. This title should be, by looking at the title, the reader should infer what is the possible content. What is the possible content in this? Like that. Okay, title should be appropriate, brief. At the same time, it should give all indication about the type of work that is contained in that. And then introduction, literature review of the current status, identifying the problems or questions to be solved should be very clearly identified, defined in the form of a question, and plausible answer you have to find out by conducting the research. And hence, from the introduction only, you have to define the objectives. So objectives of the study should be clearly defined. And here the objectives are written in the form of an aim and objective. So aim concern about the overall thing that you would like to find out. Overall, what you are achieving, that is aim. Objective means specifically what aspects you will work out to achieve that objective. So aim is the overall objective. For example, so in pharmaceutics, your aim is okay, to enhance the dissolution rate of a insoluble drug. Okay, that is the overall objective. But there are so many methods to achieve that, and hence identify which one you are going to touch. Means based upon, for this drug, based upon the literature survey, you may find that the concept of, say for example, lyophilized nanoparticles is very recent and not much information is known about this, and hence you can select okay, lyophilized particle preparation as a technique for enhancing solubility. So is it clear? The aim is broad objective, and objectives may very specifically indicating which one you would like to use. What is it you would like to test and put it to use, etc. So after this aim and objective, description of all the materials with specification. That's very, very important. So for research, pharmacy means many times we use several drugs, drug materials, excipients, polymers, etc., reagents. So their specifications are to be mentioned clearly because quality will vary from the specification. Polymers, you have to give, say for example, the molecular weight or viscosity and such other characters you have to assess, give particularly. And when you write this, okay, the specifications, you will identify the specification. You choose only those which fulfill the specification required, but test it again and confirm whether the material you got is fulfilling this requirement or not. Then when you are using it, you use it and then write in the report, this is used. So this specification is used. Like the specification of special equipment, description of the special equipment. Some routinely used equipment you need not, but if you design an equipment, certainly you have to describe it in detail. Or if you use a special equipment which is not known to many, in such a case which is very critical, you have to describe that equipment also. All other equipments, you have to identify the make, you have to identify the model, make, etc., and manufacturer. All those details you have to give with regard to the equipment. And methodologies should be described in detail, every detail, and presentation of results in tables and graphical and pictures. This is very important. The presentation of the results. So instead of giving data as such, data is the raw material for your research, and data certainly you will get a lot of data, but you have to condense it in the most appropriate form of table, graph, and picture, and present it with the help of which you can easily identify the salient character of the data. And then after giving this data figures, discuss the results. Now, discussion does not mean writing the data in words. This is like this, this is like this. No, it's not like this. Instead, what you have to do is that discuss the results, giving reasons. Everywhere you have to give justification or a possible reason and explanation otherwise, and mechanism, etc. And also try to correlate in which way your finding is fitting into the existing literature, or how it is different, in which way it is significant. Like then only you can claim that my finding is a significant one. Like that. So always in the discussion, you should give several times the cross reference. Refer to earlier work and refer it your work and their work and come up with a suitable conclusion. So discussion is very critical, and the quality of the paper depends upon how well you discussed in the scientific manner the result obtained. So more and more skill is needed in analyzing and writing the discussion part. Then conclusion with significant findings. Normally it should be conclusions also convincing with evidence, with significant conclusions only have to give, in which way it is different from the earlier one, and how it is fulfilling your objectives, how it is fulfilling your initially set objectives. Everything you have to write down here under conclusion. Then references. And references should be given properly, the uniform pattern, and all the information should be given appropriately. And abstract is needed. Okay, this is where, of course, if you want to report only, report submission means the abstract may not be needed. But for example, if you want to submit it for publication, definitely you have to write the abstract with keywords, and pictorial abstract are also needed sometimes. And even for the patents also this information is needed the same manner, and it is still exhaustive. Patent writing. Patent writing, it is more concerned about the literature, existing literature. Okay, known knowledge that should be given more in depth, but essentially the same thing you can go in detail. And so this is the way in which you have to write a report. And then the report and project proposal. So initially it is project proposal. Now it is at the end it is report writing. But project preparation of project proposal also goes like this only, adding some more details as required. In addition to this data, you need to add, okay, what are your specific requirements, in which way, substantiate them, etc. And now ultimately it is one aspect. But now how to select a research topic, how to select a research topic, that is utmost important. And especially now everyone want to work on a topic which is of contemporary interest, naturally. Otherwise, it may not be significant. So today our aim is, the immediate objective is to develop a treatment for Corona-19. Okay. So a vaccine type of product or any other type of product is like, because it is needed. It is needed now, need of the hour. Suppose you have to always do the research in the areas of current interest only. There are other ones known as thrust areas of interest. And you can select the thrust areas depending upon your specialization or interest. You can go to various sources and find out what are the thrust areas. All the funding agencies will be announcing periodically thrust areas like DVT, DSC, ICMR, etc. Related to your field, you select that. They are overall giving a guidance because they likely to fund if you submit the proposals in the same area. And hence, initially you have to overall view the subject, verify journals, interact with others, and then come up to a conclusion. Yes, I can do a research in this particular area. Then you have to improve your capacity, means expertise. Expertise is evaluated. So how to improve expertise means you have to initially do some work. So effectively some work you do it and then publish a sound article, a patent in your credit. Then it is the proof that you are having expert knowledge and skill in that area. Then if you apply, definitely you will get a fund from any of the funding agencies. So remember that, my dear teachers, that when you apply for funds, the thing that they verify is your literature, how sound you are in the literature survey, the presentation. Then how you drafted objectives, then comes what is your expertise in the chosen area in terms of publication, in terms of patents, etc. That you have, or in terms of PhD thesis, that you have to produce as evidence. And then when they satisfy, depending upon the soundness of our investigation, they easily sanction the financial aid. And hence, selecting the area and topic is at most important. Carefully you have to do, considering these aspects, plus the infrastructural facilities available in your institution. That is at most important. Without proper infrastructure, you cannot do any meaningful research. Infrastructure means 100% needed may not be available, but at least 80 to 85% infrastructure should be available in the own institution. For the remaining items, you may move to some other places, okay, centers of excellence, and get yourself trained and do the work and use it. Like that, you have to plan it. And hence, it is research. The word is simple, but it's very complex unless you do the meaningful research. Okay. And next slide, please. Next slide, please. Okay. This is regulatory considerations. Regulatory considerations are there specifically for research. It is there in different forms. What is GLP? GLP concerns with good laboratory practices. Excellent guidance is given regarding how to conduct laboratory experiments. Laboratory and animal experiments are also both are considered. Major part of this is animal, but in the same manner, GCP, clinical and clinical research and clinical trials, same principles are applicable. And then ICH guidelines are there specifically for different aspects. Different aspects. So you have to strictly follow that, and as per the guidelines only you have to. Suppose, for example, in your research, okay, there is no specific research guidelines, but suppose if you want to validate an analytical method. So ICH will give you the what are the parameters to be validated, how it should be done, etc. What specifications are there you have to follow. Similarly, in the uh, other GMP also, sometimes it is useful. Good manufacturing practices does not mean that they are related only to manufacturing, but essential. But those principles can be used in research also, in the sense that solvents, what type of solvents you can use in your research, what type of solvents you can use it in industrial production. The same concepts are applicable here also. Okay, how to handle solvents that you find in the GLP. Okay, what type of solvents you can use. Everything you find in these guidelines, and hence, though there are no specific guidelines or regulations for research, but research include all these aspects, and hence all these guidelines in a way are useful for conducting effective and meaningful research. That's why, next slide please, next slide. Okay. Now this is okay, of course. Okay. And the pharmacy teachers and students are really facing a problem over years, I observed. Okay, because I taught this subject nearly more than 34 years in Andhra University and later on in several other private colleges. And we are having this as a topic because I did a PG diploma in applied statistics in the statistics department of Andhra University and equipped well. And later on, I taught this, and we find really a problem even today also. There is no one standard textbook. There are many hundreds of books are there. Hundreds of books are there, but their orientation is in general. But when it comes to pharmaceutical, how the students are finding, even as a teacher initially, I find a lot of difficulty. Difficulty, gathered, and finally, so I thought it would be better to go for research methodologies and biostatistics. And the students, if at all they are there in the participants, now PCA as in the revised syllabus, this paper is there, research methodologies and biostatistics is there for BPharm, 8th semester, okay, and MPharm, 3rd semester is there, and of course for research scholars it is most important whether it is there in the syllabus or not. And but surprisingly, what happened is the PCI, the syllabus, this is made as a non-university paper, non-university exams. Unless the university examination is there, the faculty and student may not be taking real interest. Of course, it is useful. They realize only after completing the course. When they join somewhere in a research organization process, or when they join industry, they realize what is this. But okay, it is there, but because it is made as a non-university question, paper and examination, so teachers and students started neglecting. So why I'm referring is, please do not neglect this, and you should have this paper full. PharmD also it is there, but PharmD examination is there. Okay? Only BPharm, MPharm. It is non-university examination prescribed, and started giving negligence. So try to be more serious. And if you want to establish a good career as a researcher, this is vital. This is vital. Okay, this is vital, of course. Okay, in planning, it is vital. And certainly earlier I know that the research scholars and teachers, they used to go to statistician for getting information related to their experiments, planning, etc. analysis. Now we can do it. All the students are there. Optimization. I still remember for several years we used to run to the statistics department to analyze, to use the optimization technique for analyzing data. Okay. So now there is no problem. So with that idea, to make it simple, this is an attempted, and this topic is very much interesting to all those who are interested in research. Okay. And this is an insight into the basic concepts of research, but research is vast and multidisciplinary. So you need the knowledge, essentially knowledge and skill in your own subject. But this is how to conduct the experiments meaningfully and how to derive the data such that data should be reliable with the help of which you can make out meaningful interpretation. That is the object of statistics. And statistics are very vital for successful research. Okay, without application of statistical principles today, it's not possible to turn out a good piece of research work. Okay, so with this view, I thank once again. Conclusion. I thank Dr. Jayabalan and the management of Alwar College of Pharmacy for giving me this opportunity to meet you all through this online program. Thank you all once again. Wish you all good luck.
Thank you very much, sir. Thanks for your elaborate talk. Now I request Professor R.K. Goyal, sir, Hon'ble Vice Chancellor, to conclude the session.
Okay, so actually the time is too much. So, but there were I read the 3 questions.
Okay.
First thing I would like to say, I must compliment Dr. Chowdary for giving such an exhaustive talk. I will describe this talk like a big spatule. Spatule means the one which has got several ingredients in the one capsule. And so sir has included not only the research methodology, but the statistics, biostatistics, the clinical research statistics, QBD, directly or indirectly, the factorial design, and then going beyond the regulations required, like even the GLP to all that, and then going again beyond that, how to write the report, what to do. I think there cannot be, I mean, it's something like if one has to take it in to the fresh students, it may take maybe at least a week. I mean, it comes minimum 7 to 8 lectures. That is minimum. I mean, that is also very comprehensive lecture. So this is a worth compliments, and I really admire that how you could do this much.
Thank you, sir.
Questions, if you feel like I can answer directly, because sir must be exhausted also so much. Number one is, somebody has asked that what are the source of statistics? Now I have to answer this question. Sir has given the book, but let me tell you, my only answer is one should not be the one who is teaching the statistics should not be somebody from basic science and teaching you the biostatistics or statistics. This is, it's a rather, you will be simply lost in the a sea of data and all that. Ask, I mean, the best source is the one who has really done the research,
Yes.
Chowdary, or somebody who has done the research, and they tell that these are the statistical you should know. One should not become mechanistic. This is my only answer I can say. There is no, there are books available. I used to read that Mahajan simple, but it was not from some pharmacy point of view also. But if you get the MBA called for such lectures of biostatistics or statistics, I tell you are not going to do justice. I have done this for the doctors also when I was in Ahmedabad. They used to say, sir, it is so simple. The way in which you have talked in 2 hours is so simple. So that's what has to be there. This is one answer to my, for somebody who has asked the question number 1. Question number 2, it has 2 parts. One is, of course, sir has already answered. That was if there are several factors, how to do it. Probably this question was before that. So the best is quality by design or the factorial design, which are supposed to be there. And the basic principle is regression analysis. In a very crisp language, I can say, if you understand regression, I mean correlation, regression analysis, and then the factorial design will be easy. It has to be sequence. If directly you want to learn the QBD or like that, nothing will come.
The QBD and optimization are based upon only factorial design and regression analysis. That multiple regression analysis of
factorial design and regression, and it has to be systematically.
Yes.
The basic you cannot understand. Let me tell you that also. And don't go by those programs also of the statistical programs of the computer. No, you have to understand the basics.
Basic.
Teacher, try to teach that first and then you go for it. So this is the first part, and second part was about the use of the same thing in continuation, the artificial intelligence or multiple.
So that is also there.
That is all, it will come.
All everywhere.
You will be in a position to analyze. What we say artificial intelligence or the neuronal network and all that, all these things will come. So this is what is the second important question which was asked by somebody. Somebody asked one question. The answer is yes for the second part. That can be that I don't remember. I've noted down, but I will
artificial intelligence.
Yeah, research development mostly confined to government funding. Sorry, this is a different.
Yes.
See, there is nothing like that you have to go to CSIR labs or like that. If in the lab, even in the limited resources, if your idea is good, if you follow the principles what Dr. Chowdary has given, you need not to go. They will rather have to come to you, professors like him, to get the ideas, and they should collaborate rather than we should be dependent on them. This is what is there. So that is the thing which I think these are the answers to. So sir, you can add if you want anything in that. Yes, sir. These are the questions.
What you said is exactly correct, sir. What you said is exactly correct, and I appreciate the interest you have shown in following during the last 2 hours this program. Normally the topics like this take more time. Instead, if the topic is selected related to a particular research I am doing, it may not be suitable for all because it's teachers and students are very heterogeneous. Their interests are varied, and hence first I thought better to give an insight into this research proposals, etc. Any subject they can do from. And what you said is appreciate the participants shown interest in optimization and the QBD. In fact, I thought of first doing that, but it will be useful only to certain group of teachers and students only, not for all. So as I said, the QBD, the term is new, but inherent concept is factorial design only, factorial design and multiple regression analysis. And when the factors are 2 or 3 or 4, we can do it manually without any software or computer. But when the factor size is increased normally, we need the software. The principle remains the same, QBD also. They're almost same, everything. Similarly, optimization, there are so many optimization techniques are there, but inherently they're all based upon the same concept. And in fact, the optimization and QBD, the students and teachers could not exactly follow what the people are telling from industry. That is also there because several of my students are working in industry and they approach me. They approach me, sir, it is like this, our boss is telling like this, what to do. I clarified several times. But here, this is specific. The student and teacher should sit together and take some data, case study, and then implement. Otherwise, it's very difficult. Speaking from a steady, from mic over mic and a slide, the participant cannot understand fully. It's what is needed is to these techniques like pharmacokinetics, the same way, the teacher and student should sit together and workshop has to be conducted. 2 into 2 interaction should be there. Then only knowledge can be transferred effectively. Otherwise, it's not possible. It's a wonderful idea. This I suggest Jayabalan may organize such a workshop. If at all you want to put it to more use or useful transfer of knowledge, a workshop is better in certain areas like this one, optimization QBD.
Sure. We'll try to have one. But we are admiring, and it is an amazing thing. One who really loves the teaching can continuously talk for 2 hours on a topic without even drinking a drop of water. This is an amazing thing, sir. You are.
Sir, Because
we love
this
you
is
all.
as a teacher, almost as a teacher, every teacher
Sir,
is
we have
equipped like
among
that,
us
maintained like that.
two eminent teachers, so those who are really loving the excellent teaching, and they're doing it. You are
Thank you,
doing
sir.
it very perfectly, and thank you very much, sir, for being with us on this Saturday. And I once again thank Dr. R.K. Goyal, sir, for sparing his valuable time on Saturday. He sat with us for 2 hours continuously.
Surprised.
I am very thankful to you, sir, and I'll whenever I personally meet, I'll get your blessings. So both of you, from both of you, you are all real teachers who are really loving teaching. You have teaching in your blood. But I don't have any words to say more and more about you people, like those who are loving real teaching. So this is a very encyclopedic presentation you have given upon research methodology and biostatistics, including basics in QBD. So this is very amazing, and we are highly impressed, admired, and we are thankful to you, sir, Professor KPR Chowdary, sir.
Thank you all.
I would like to hear more and more from you. And as Dr. R.K. Goyal was saying, this is a matter for 7-8 theory classes, condensed and presented in a pleasant manner to the people within 2 hours. This is very great thing. You have done an excellent
Thank you, sir.
job, sir. You justified it, sir, with your presentation as well as your book. Thank you very much, sir.
Thank you,
And
sir.
I thank once again Professor R.K. Goyal, sir, for sparing his valuable time, sir. We have taken your Saturday on the whole. I thought I am going to take only 1 hour, but it is almost over now. Thank you very much, sir.
Thank you, sir.
3
Thank you,
hours.
Goyal sir. Goyal sir, thank you very much. Thank you, sir. Nice.
Thank you, sir. With the permission of the
Yes,
chair,
I-
I would like to end up the session with.
So it is wonderful that we both shared the same platform. Goyal sir.
Yes.
Very nice
I
one.
also enjoyed a lot and very nice. So nice. Thank you, sir.
Thank you very much, sir. Thank you.
Thank you, sir.
I don't have any words to express my thanks, but really,
This is the
I
routine.
mean it. Thank you. Thank
We
you
have
both
to
of you.
thank you. You are providing a platform for
Sir.
all of us. Thank you all.
Sir, thank you.
Thank you. Okay.